Right, okay, so we're gonna carry on from this point. So where we finish with metaphase, we align those pairs of chromosomes. So the next logical thing is to draw them apart, right, which is going to be anaphase. So let me again free up a bit of space. That's all. So anaphase 1. So what do you think? How shall we phrase it? What do you think is getting drawn apart?
Chromosomes.
Chromosomes, okay. Out of what? What do you start with? Out of pairs of chromosomes. So can you see the difference? In the metaphase of mitosis, we were drawing apart, what were we drawing apart there? Sister chromatids, right? Sister chromatids. So we were drawing apart those sections, but here we're drawing apart the full chromosomes. Yeah. So each chromosome. So this picture, so it's blue and green one are going to be drawn, and then red and black ones are going to get drawn to the other side. Okay? And this, you remember what I said, because of the different outcomes, here the outcome would be red and green would be on one end, blue and black would be on the other end. So let's stick with this picture and carry on from there, so we don't get two lots of pictures going on at the same time. So in telophase, right, when we're going to start getting the cells split, yeah.
Just, do you know on the anaphase?
Yeah.
So the blue and the green are both going to left hand
So
side,
they're
and the red and
gonna,
the black
so
are going to
blue
the
and
right.
green are going to be moving towards that end,
Yeah.
that pole. The red and the black are going to be
towards
going
that pole.
towards the other pole. So if you don't mind, I'm not going to draw another
No,
separate
that's I am,
diagram
because I just
for
want to understand
anaphase,
it.
but if you wanted to, you could. So then in telophase, so I'll put sort of telophase and cytokinesis into one one, because I've already started showing that that cytoplasm is getting pinched. So telophase and cyto. So what would normally be happening in telophase? Again, from mitosis, what would you typically associate with telophase?
The spindles have disappeared.
Right, spindle disappears. Right, so spindle. What else is happening? Somebody's saying something about nucleus. Right, so nucleus appearing. How many? Is it just one nucleus, two nuclei?
Two.
Just two. Right, so two.
And it's split in half the cytoplasm.
And then the cytokinesis part of it is going to be splitting of the cytoplasm. So, if I draw my two new nuclear membranes, can you draw for me the kind of what are going to be finding inside? Can you draw the right colors of chromosomes found in each nucleus? And then I'll go and have a quick look. So what's going to be the end result?
Would it be the two that's been drawn to each side of the pole?
Yeah, exactly, because then you're going to do is squeeze that midsection to split two cells and to split that original cell into two. Okay, so let me see how you get to. So good, yeah, okay, that looks
I haven't
right.
drawn it, but it's new, so I'm just trying to look into the things I can get.
Yeah, so can you draw chromosomes, the resulting chromosomes inside? And I'm going to just have a look, see if you've got it right. Yeah, okay. Perfect.
Blue and
So do you get the logic, right? So whatever you got here, that's going to end up in one cell, and the other two are going to end up in the other cell.
this one.
Yeah, okay. I could have given you, I mean, you could put the colors, yeah?
I'm very lazy.
Yeah, okay, right. So I think
And
people who were drawing did get it right. So with my drawing, in one cell I would end up with a blue.
but it would be like what's it called? Do you know how you said they would? Sorry for that now.
Yep.
That would be crossed over though, right?
Yes, they would be crossed over. So you remember what I said? It's just to not overcomplicate it. There's already so many colors and things going on. I'm just disregarding the fact that they split, that they exchange some things. Yeah? During crossing over. So then this one is going to have black one and okay? And what also could happen is that those chromosomes might decondense, right? So you remember at the moment I'm drawing them as fully condensed, but in reality they might start unravel a little bit, okay? So chromosomes
Creating like a different DNA.
decondense. But equally they might still condense, because you remember, is this the end of the whole thing? No, because we still have the second division going on next, right? So in some cells you might have no decondensation happening and the chromosomes just stays there. Right? And at this point you're going to end up with two separate cells at the end of cytokinesis. Right? Obviously that's going to keep getting pinched and pinched and pinched and pinched more, and then we end up with two cells. So let's think about the number of chromosomes in these resulting cells. So in my picture, remember I started with a cell which contained 4 chromosomes, right? Just for illustration purposes. So how many chromosomes have I ended up with now on my picture? Just 2, right? So I've gone from 4 to 2, but in reality you're going to go from 46 to 23. But the only issue is, and that's what Chris was asking early on, well, why do we bother then having a second division? Why not just leave it? Because we already seemingly achieved what we wanted to achieve, i.e. half a chromosome number, and we also exchange those fragments and arrange them in random ways to achieve genetic variation. The only issue we have is each chromosome still have 2 sister chromosomes, two lots of DNA, and I need 1 lot of DNA. And that's why you have to then do second division to still separate those sister chromatids and get that bit sorted. Okay? But we're going to stop here, and we're going to talk about the second division another time. Okay?